kind of launching right in here. This is one of the fields that we worked in with the Soil Health Initiative, Minnesota Soil Health Initiative at NRCS. So that was a 5-year agreement that farmers made to plant at least 2 species of cover crops in the fall. That meant that you weren't having fall tillage in your field. And you did it in the same acreage every year. So this is a cornfield, location not to be disclosed. One sweet corn field, actually. So one side of it, the corn had been harvested, it had been ripped out. Then they tilled very aggressively. You can barely see right about here, a corn root system left in the soil. Otherwise, not very much going on that side. And on the other side, we had the corn root systems, which had kind of been yanked out a little bit when they were planting the cover crops, and 4 or 5 species of cover crops in there, some that had been seeded that year, some that were volunteering. And it was just this night and day kind of comparison between the two. And so what I would like to do, what I got asked to do, I'll go back to that field later, is kind of give you an overview quickly of soil health, a little bit about soil biology. And then I thought, might as well do some tie-in for aquatic systems as well, and the implications of soil health. So the first thing is, what is soil? And the official definition includes the following elements. It's a natural body. It's composed of solids, so that's the minerals fraction and the organic matter that you can see there. Liquids, usually water, but not restricted, and gases, primarily air. It occurs on the land surface. It occupies space. And here's where it gets really distinct for what makes it soil. It has layers that are different from the parent material being bedrock, due to the addition, loss, transformation of energy and matter, usually through a combination of chemical, physical, and biological processes, and or the addition to support rooted plants in a natural environment. So I'm sitting here in my sunroom. I have a great big grapefruit tree kind of staring at me because it's growing in this pot of pathetic medium that it would really prefer that I change out. That is not soil, that potting medium. It is medium. And when you look at the graphic on the right, where you see the pore spaces and everything, that pore space is really unique. So in soil, you have the interaction of the physical land, the water, and the air on this microscopic scale, and it's all around you. And the number of processes that are going on in those tiny little spaces is incredible. We can't even understand all of them yet. We're just getting to that point. So all of those processes pretty much are driven by the soil organic matter. So if you look over to the left, anything living or once living in soil is organic matter. So that's another one of our fields. You've got some manure, you've got some corn debris, there's an anthill in there. All of those things are soil organic matter. So you might have 20% in a temperate forest soil. Prairie soils, our native prairie soils remnant have about 10%. So 90% of soil function is depending on that organic matter. And you find that organic matter principally in those soil pore spaces. So keep that in mind as we're going. Because those soil pore spaces, I have to get rid of some pages here, are very vulnerable to our action. So going back to the soil forming in layers or horizons, on the right, there's a USDA graphic. On the left is a picture I took in Mower County. Honest to God, I did not make that graphic look like that picture. It just worked out that way. So you have the O horizon on the top. That's your duff, your decomposing organic matter. It's the thatch on your lawn. It's the leaves in the forest. That's the stuff that's being actively broken down and then incorporated into the topsoil. So topsoil is really what rock and roll is about soil. And it's the stuff that we're losing in Minnesota particularly, but all over the country, in fact, all over the world. Topsoil is the ultimate endangered species. And that's where most everything lives, is in the topsoil. So if you have something like this, if you dig a hole in your backyard and you see that going from darker brown where the organic matter is into something lighter, you're getting into things that are progressively more mineral in nature. And that means that they're progressively less lively. It does not mean that they're completely dead. It just means that they're less lively. So in soil, that A horizon, thinking about that, that's the part we use. That's the part we need to protect. Also, soil can be in waters. So if you just put a layer of water on top of this picture mentally, that water could be 2.5 meters deep. As long as there are rooted plants, not kelp with holdfasts, but rooted plants growing in it, the bottom of that lake, that stream, that wetland, that's soil. It's not sediment. So if you only have the inorganic 5, the 5 components being sand, silt, clay, air, water, so far we're inorganic, and then organic matter, what you have is dirt, because nothing's alive in it. So if people have seen the movie The Martian, or they've read the book The Martian, they might remember that the stranded astronaut Watney, who was a botanist, decided that he could use frozen poo to grow potatoes. It was the great aha moment to take Martian soil and turn it into something that he could survive with by actually farming. And there was a slight problem there. And when the book was written, when the movie was made, we didn't know what the slight problem was. So we've been trying to model Mars soils, and there have been a couple of experiments where they planted various lettuce and then Arabidopsis, which is kind of the all-purpose, it's the Drosophila of plants. You do all your research on it. It's a wort. And they had 3 media. They had fake Mars soils that were mined. They were geologically similar. One in Hawaii, the other site was in the Mojave Desert. And then they formulated a Mars soil from volcanic rock and clays and silts and chemicals that the Curiosity rover had discovered. This on the slide is the Perseverance rover being lower down, much more capable rover than Curiosity. And both plants sprouted and grew in the mined Mars-like soil as long as you fertilized it. Because you see, this was sterile. So there weren't any microorganisms there to provide that nutrient cycling that's so important to plants. Neither of them grew in the formulated dirt, even as transplants. So what we were seeing was that formulated dirt was a little bit off. And part of the reason is that it had a pH of 9.5. So that's about like caustic soda. That's way high. And it was a pH of 7, so close to neutral in the mined material. So they tried adding hydrogen, H2SO4, so sulfuric acid. They got the pH down to 7.2 in the formulated material, and they could keep transplants alive in it briefly. So already this is a problem. So making soil is tough. Now it's tougher than we thought, because Curiosity, after this experiment had started, documented that a salt called calcium perchlorate accounted for about 2% of the surface of Mars, where Curiosity was testing. And when you added that to the formulated soil, everything died. The plants just did not grow in it. Now, it just so happens that on Earth here, we have these bacteria that eat calcium perchlorate, and then they give off oxygen. So they're probably part of the group that actually terraformed Earth and made the surface plant-friendly, plant-capable. So one of the questions that I saw reading the various comments was, well, why don't we just ship these bacteria to Mars? is transplanting them would be a terraforming win-win. You know, they'd release oxygen, they'd restore the atmosphere. Well, they do need water to do that. That's kind of a problem. And it just kind of brings back around this idea that there is no planet B. We need to take care of planet A, and specifically, we need to take care of the soil on planet A. So the next idea is, what is soil health? I said I'd be returning to that field. So these two clods were collected, one from the highly tilled part on the left and the other from the not as aggressively tilled, although it does get spring tillage, and very well planted, very busy soil on the right. And it should be pretty obvious that there are some extraordinarily important visual differences between these two. So there's a quote in law that Justice Potter Stewart in 1964, he was ruling in a case that had nothing to do with soil health. But the quote that really sticks out there, people use it a lot. I know it when I see it. Okay, I know what soil health is when I see it. Or in this case, also when I smell it. The definition is, it's the capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. Okay, the soil on the left is not sustaining anything because nothing is allowed to live in it right now. The soil on the right, you've got roots of grasses, you've got fungal hyphae. When we were working in that field, it was the second week of November in 2016, that record growing season. We had all kinds of insects walking around, flying around. There were plenty of earthworms in there. When you broke the soil open, it had this rich, earthy smell to it. And the only real structure in the one on the left is a macropore that was created by a corn root. The previous year, that root had been pulled out of the ground and it left that hole behind it. So in theory, we could have had some kind of colonization of that space, but we saw no effort or no evidence of it in the few hours that we were in these two fields doing our assessments. So what this is a demonstration of having a rhizosphere, which is the soil within an inch of a root, and not having a rhizosphere. So we have plenty of soil within an inch of the root on the right. That's the place where all the biological activity is, because the root is busy chucking out root exudates, sugars, alcohols, there's some small lipids, proteins, and it's busy feeding all of those microbes that are its partners in the soil. And then they're busy recycling that amongst themselves. There's a whole ecosystem with multiple tiers, predators, prey, parasites, all in that rhizosphere. If you look at it, the left-hand clod looks a lot like a food desert because there really isn't any space there that's around the root. The right-hand clod is busy and lively. And in addition to just being able to see the plants, seeing the fungal hyphae, the other thing that's going on here is those fungi, at least some of them, are producing glomalin. And I call glomalin the dark carbon of the soil universe. It's recently discovered. It holds the sand, silt, and clay together in stable aggregates, along with the little root hairs. So not only is the soil on the right more lively, the soil on the right is more structurally stable than the soil on the left. And then if you also look at them, those two clods came from about the top 8 inches of the soil. In the top 8 inches of the soil, you see most of the fungi, you see most of the roots, most of the soil organic matter is in the top 4 inches. So if you think about how we handle soil, how we treat it, we're putting all of that activity at risk. And just to give you kind of an idea of how much activity we're really talking about, roots are funding this system or fueling this system, depending on how you want to envision it. All of these organisms, including in some stream banks, fish, are working with the environment that plant roots are creating. So plant roots and the bacteria and the fungi around them are the base. Above that, you get everything else. So tardigrades, water bears, people are familiar with them. You've got all the microorganisms. You do have some primary producers there because there are some algae in soil. But basically, those microorganisms are there driving all the cycles. The water cycle, the carbon cycle, nitrogen, phosphorus, they're just moving that stuff around. The multicellular fungi can be thought of as mutualists, the mycorrhizae, are actually living with and in plants and hooking plants together. So plants can communicate through this internet of soil things with one another. So they can say, oh, I'm running out of water over here. You might want to be careful how you use water over there. Or I'm being chewed on by an insect. Maybe you want to get ready to be chewed on. They're sending these chemical messages across. The saprophytes are the fungi that are decomposing last year's and previous organic matter. And then there are some parasites and some pathogens in there because this is a complete little ecosystem. There's another complete ecosystem of nematodes. You've got shredders, you've got parasites, you've got predators. There's another one. If you go and you look at mites, again, these multiple layers of complexity in the soil, it's an incredible community. You can have up to 20,000 pounds of organisms in a single acre in the top 6 inches of healthy soil. 1 teaspoon of that soil could contain up to 10 billion microbes, representing 11,000 species. So I do try to tell people, if you're out picnicking and you drop your sandwich, leave it, because it is soil organic matter now. You should just let the microbes have it. Otherwise, they might decide that you are soil organic matter when you eat that sandwich. Of the poundage of organisms in that top 6 inches, most of them in terms of biomass are going to be earthworms. This is a double-edged sword here for us because we don't have a lot of native earthworms. So earthworms have been very successful invaders of previously glaciated land. Right now, we have a new one coming, the Asian jumping worm, which worries me deeply because they can take perfectly good soil and make it into nasty little coffee grounds. In 1952, and I've had a really hard time finding studies that say how many critters live in the soil. There was a study done in the Duke Forest in North Carolina. They estimated 124 million arthropods per acre. That included 90 million mites, so that ecosystem of mites I talked about. 28 million springtails. You might see those if you're digging. You might see little white things pop up. Those are springtails. And only 4.5 million other insects. So when we're talking about populations of insects potentially crashing globally, numbers-wise, every insect kind of might count. And I'll tell you later how important soil is to insects. In farmland, the study also done in the 1950s, they were estimating 425 million arthropods. Again, arthropods, paired jointed appendages, hard exoskeleton, everything from mites up through spiders, insects, lobsters, crayfish, they're all arthropods, of which 209 million were mites. So that's a critical part of our soil ecosystem. Now, returning to earthworms for a moment, because they do actually do some great stuff in our soil now that they've recolonized it. So bioturbation, especially when we're thinking of urban soils, formerly agricultural soils, currently agricultural soils, is how you move soil the natural way. So building structure one earthworm at a time, quite literally, including those jumping worms. There are 3 different life histories of earthworms. You've got epigeic, they're the ones up in the duff layers. So right above that blue arrow on the left hand is where you'd find the epigeic, the endogeic. So epigeic on top of the soil, endogeic in the soil, tend to live near the surface. These are the 2 that actually are the most vulnerable to tillage because they're up where tillage implements are. They're also the most problematic in woodlands because these are the groups that are turning that duff layer in, and our woodlands are adapted to having that duff layer there. So that's a bit of a problem. The anisic worms are the ones, the night crawlers, they make these vertical tunnels that can be 2 meters down. And they have this really interesting lifestyle. They climb up, they grab a hold of a leaf. So it could be a corn leaf in a cornfield. They'll just pull that thing right down into the soil. And then they'll eat it when they get down to the chamber at the bottom of their tunnel. So they are physically incorporating organic matter quite deeply into the soil, as well as creating that tunnel. If you look down in the center, you'll see an area of slightly lighter soil surrounded by darker soil. That was an earthworm tunnel vertically coming up from a lighter colored horizon, one of those B horizons, sandier looking, into an A horizon on one of the farms that we looked at. And because earthworms are eating while they're moving, so you can think of an earthworm as a conveyor. What goes into the mouth comes out of the back end, and they eat everything. So they are literally ingesting soil. They will eat, they will gain nutrition from the organic matter in the soil, but then they're repackaging that as their cast, which contains the soil from where they were, plus their digested organic matter, which is now even more finely broken up and more available for the bacteria to then recycle. And then a certain amount of slime that they make to hold their cast together. And then that ends up packing the inside of the tunnel behind them. So you can see the movement of soil very clearly there. On the right-hand one, where we have these scans of soil cores, you can see root channels are fairly small, but the earthworm burrow, and these are from the anisic worms, the night crawlers, are really quite large. So the earthworm tunnel that goes straight up and down becomes a pipeline for water going into the soil when it rains, for air, which gets sucked down into the soil as water passes into it. And then for plant roots, if they happen to encounter one of these earthworm tunnels, you can imagine it's self-watering, it's aerated, there's fertilizer on the side of the tunnel, and you don't have to do any work to extend other than the work of growing. So the earthworms are in our soils an important engineer that's working there all the time, not the only one. So if we have some things about soil, all these cycles, all this life that's going on, and we need to protect it, NRCS has the soil health practices that are recommended. First one is minimizing disturbance. So disturbance means either tillage or traffic. That traffic can be foot, can be animal, it can be wheel. And basically you just want to minimize that as much as you can. So if you're tilling a soil, eventually you're going to form a subsurface compaction layer because you just keep going down to the same depth and stirring the soil, and you end up with what's called a plow pan at the bottom of that depth. And that compacted layer can impede root growth, invertebrate movement. Earthworms can't necessarily get through it, and therefore also water and gas exchange in the soil. And my colleague here, and this is that same field that we had on the beginning, is showing a tillage radish. And these are called tillage radishes because they poke these gigantic holes in soil. But even though that root could exert 300 pounds per square inch, and that's radially, not longitudinally, that's a lesser amount of pressure that the root can put on, it could not get through that compacted layer. We could hit it with a shovel. We knew it was there. And if you look at the root, you'll see that my colleague's hand is holding a part of the root where there's a lot of little branching and it's dirty. And then farther up the radish, it's clean. Well, the radish couldn't get through the compacted zone, so it grew out of the soil as it continued to soak up all the excess nitrogen and phosphorus from this heavily fertilized soil. And when it was without the support of the soil, it fell over. So that's the first bend. And then that put its leaves in the shade of the other plants. So then it bent the top back up. That's called positive phototropism, so that it could get its leaves back in the light and keep photosynthesizing. So that is about as good a demonstration of what compaction means in terms of plant growth as I can imagine. And these fields were not hit with, if you look at the bottom, the tillage disturbance profile, a moldboard plow. They were hit with something more like a subsoil shank, but still that layer had formed and this radish couldn't penetrate it. So if you really don't want to disturb the soil much, you would, and you have to plant something, cover crops, a prairie seeding, you know, anything like that. A no-till drill and knife is only going to disturb about the top ½ inch and only about a ½ inch wide. And those boxes represent standard corn row spacing. So 30 inches across, and then I had them go to the depth of the subsoil shank, which would be 14 inches deep. So you can see how much less soil, if you're trying really hard and you need to plant something, you can disturb if you use the no-till drill and the knife. And then thinking about soil that used to be agriculture. So a lot of our suburban areas are on former farm fields. Even one-time damage in these environments can linger. So we have a couple more, I think these are CT scans of soil cores. They come from a Danish sugar beet field. Sugar beets are incredibly hard on soil. You have to till very aggressively. You're pulling the beet out of the soil, you're turning the soil out to get the beet. And so the cores that were put in the scanner here were taken from, they're both 8 inches high, 8 inches diameter, taken from 10 to 18 inches below the surface. The one on the left had never been driven on. The one on the right had been driven on by a beet harvester, which is a pretty heavy piece of equipment, once. So if you look at the amount of damage and you see all the pores, as in the previous slide, that are created by roots, by earthworms, you can see how much of that pore structure had been lost. It had been crushed. Now, this isn't driven over once and we went and dug it up and looked at it right then. It had been driven over once 14 years before the soil was dug up and looked at. So earthworms can move soil, plants can move soil, they can create tunnels, they can do this work of creating that home for everybody to live in, but it takes time. And you can see how much of that physical structure was lost because of disturbance. And disturbance, you know, you don't have to be on a farm, you can be in a backyard. I include this also because I've seen enough rototillers, even in my neighborhood in Minneapolis, I admit, full admission, to get rid of surface compaction in my tiny backyard due to 4 sons and 3 dogs. We brought in a rototiller to break up the crust on the surface. That is the only time my yard has seen that, and I've owned this house for 30 years. But when you think about a tiller, especially a rototiller, when people are using these in small landscape applications, basically what they're doing is they're taking their soil and they're putting it in a blender and they're putting it on frappe. And so they are not only destroying the structure that's there, they're literally emulsifying everything that lived in there. So roots, fungal hyphae, everything is getting thrown around at that kind of speed. And then it sits back down and it'll want to collapse. And if you did that because you were going to mix in your compost, which is in the middle picture, what you're also doing is you're throwing all that soil into the air and you're adding oxygen to it. So you will get that burp of CO2 when the microbes get to work on the organic matter that is all of a sudden absolutely full of oxygen. And then you've kind of diminished the impact of adding organic matter to it because organic matter is getting oxidized by the bacteria because you just mixed everything up. Soil health practice number 2, increase plant diversity. I really love this image on the left. This was a PhD thesis in 1919. I'm pretty sure that Mr. Weaver went out and dug everything up with a spoon so that he could get those beautiful pictures of the roots. And those are native plants. And you see all these different depths of rooting. And if you look, there's a scale on the left is in feet. So some of these native plants are getting down 6 to 8 feet. Corn and soybeans will get down to 6 feet. Alfalfa will get to 20. But I circled P. P is POA. And POA pratensis is bluegrass. It's what we put in our yards, and it only gets to 6 inches. So if you think about a native environment where you've got a lot of different plants, all those different plants have different root architectures. And they have tap roots, they have fibrous roots, they're all producing different root exudates, they're depositing organic matter from the roots at various depths of the soil, and they're creating this web of food in the soil at all these different depths for all the other organisms to make use of in the soil. Now, when we go and we say, oh, well, we're going to plant all grass, or we're going to plant all corn, then we're doing the exact opposite. We're only adding root exudates, which can be 21% of all the photosynthate, that a plant produces. So 21% of the fixed carbon. But we're going to put all of that just at one layer in the soil. And if we're in a field, a row crop field, even worse, we're just going to concentrate it in one stripe going down the field. So we're going to create food deserts horizontally at various different levels because we're not feeding the soil. And then we're going to possibly create them as well with the direction of our planting if we've got bare soil in between. So when I talk about the associations, plants have preferred partners that they work with. They will actually seek them. They will put exudates out trying to call the bacteria that are the best partners for them in terms of cycling nutrients. And a really good example that I learned about at the Nobel Conference at Gustavus, the 54th one, is big blue stem, which is one of our dominant prairie grasses. It's also been looked at as a bioenergy crop. And it has one particular bacterium that's been discovered called Verrucomicrobia. And it grows much, much better in the presence of Verrucomicrobia. But if Verrucomicrobia is not in the soil, the plant becomes much more cold sensitive, and it has more drought sensitivity. This should probably explain why the big blue stem that I put in my yard from a pot didn't do well over time, because I probably don't have any of that bacteria in my soil anymore. But what was also interesting is that you can't culture that bacterium well in a lab. So when people are saying, oh, here's a bottle of soil health, here's all the bacteria that you need, all the fungi that you need, you just add this to your soil and it will be great. They don't know, because they don't know what the plant might actually need. And they might not be able to culture what the plant might actually need. But if you go in and you add more plants, you increase that diversity, you might revive some of these partnerships, because a lot of our soil microbes are able to wait for a long time for their opportunity. Another one, keep the soil covered. This has two aspects to it. This is really easy. Keep it covered with anything, mulch, leaves, plants, anything. The first reason is because soil, when it is not covered, cooks. On the left, we have a couple of thermometers, both at noon in Nebraska cornfields. One had cover crops, the other didn't. The one that did not have cover crops at noon, the soil is already 107 degrees. And that's noon. You still have all the afternoon heating to come. And once you get to 113 degrees, those soil bacteria are going to start dying. If you look at the right, that's one of my research cornfields down at Rosemount. I was monitoring soil temperatures so that I could model corn rootworm development of the larvae. I only cared during the period that corn rootworms might be larvae. And so that I stopped in June because they hit third instar, I was done. But if you look toward the right side of that, you see there are 3 days where in the pre-emergence herbicide field that I was using, and boy, that pre-emerge looked great. There wasn't a thing growing in there other than corn. That soil was absolutely cooking in the middle of the day. And then at night, it even got colder. The green markers are a field, a little plot right next to it, where I had a lot of grass growing in between the corn, and that moderated the soil temperature. Now, part of the reason, other than the soil bacteria, that you'd be interested in soil temperature is that roots get stressed out by extreme temperatures as well. So plants do most of their photosynthesis, or do photosynthesizing rather, most efficiently between 77 and 95 degrees Fahrenheit. So on a hot day in the summer, when you go and stand under a tree and it just feels so much cooler, you almost can't account for it because of the shade alone. Well, that's because the plant is air conditioning itself. It's taking water from the soil, it's transpiring up to the leaves, it's throwing it out through the stomata, the openings on the leaf where gases and water are exchanged. If the water evaporates and the evaporative cooling cools not only the surface of the leaf, but the air around it. And so you feel that. Well, in the meantime, in the soil, you have now had to remove water to make that work above ground. When soil gets drier, it warms more easily because the specific heat of water is greater than the specific heat of the mineral parts of soil. So on that hot day, an uncovered soil is not only going to heat directly, it's going to be kind of getting that extra bump of heating because it's getting dried out by plants that are desperately trying to keep themselves cool. And then their roots are getting stressed by the temperature of the soil around them. So it's a win-win-win if you keep your soil covered on a hot day. But soil also needs armor. This is a single raindrop hitting a bare soil. If you look at it carefully, in the splash, you can see dark things. Well, those dark things are the sand, silt, and clay of soil, of those little aggregates that are so important in soil because roots hold on to aggregates in the soil. They are being rain bombed into sand, silt, clay, and sludge because that individual raindrop can travel at 20 miles an hour. That's the terminal velocity of rain. So think about a low-speed car collision, how much damage a 20-mile-an-hour collision can do. When that happens, you've now blasted the soil apart and it can be thrown 5 feet in just about any direction. So now you not only have the aggregates getting broken down, things are getting thrown all over the place. They could be thrown against a building, they can be thrown out of a field, but they're not where they need to be. So if you have a well-armored soil, this well-armored soil picture is Gabe Brown's ranch in North Dakota. Gabe Brown is an early soil health adopter, and he has got his place cooking but good. If you look in the middle, you'll see there are green lines. Those are his crop plants, but in between is all of his cover crops. And he'd been no-tilling for years by the time this picture was taken. This picture was taken the morning after a 13-inch rainfall. Where are the puddles? Where is the erosion? The picture on the right of bare soil, that is a 4-inch rainfall destroying a cornfield in Iowa. Now, 4-inch rainfalls, if you look down at the bottom left again, we're starting to see mega rain events are even more than 4 inches. So if mega is becoming more common, 4 inches are becoming just about, every day. Well, not quite, but 4-inch rainfall events are becoming really very common. And that lower right picture is one of our soil health initiative fields after a 4-inch rain. The 4-inch rain had occurred very shortly after the field was harvested. We went down to sample that field and its cover crop partner about 5 days after the rainfall. The cover crop partner was still pretty wet, but we could work in it. We couldn't walk in this one at all. We had to wait for it to dry. We waited 3 weeks for it to dry. So comparing the two fields became very difficult. In the meantime, the soil, you can see that crusting. That's what happens after the rain bomb effect. So the light part is the silt and the clay in the field that had sealed the top of the soil and then cracked. So now this soil can have water and air go in through the soil cracks, but the surface of it is like cement. So in keeping your soil covered, you're also protecting against that kind of damage. And then another part of it is when you get that kind of damage, you're getting the runoff. And here's where I'll talk more specifically about soil health and water. What we have found is that a healthy soil is not only able to take in water more easily, it doesn't get blown apart. When it gets blown apart, and as I mentioned, the silt and the clay and the sand is now moving out of the field, one of the things that's moving with it is everything that's been applied to that soil. So in terms of city soils, what we're looking at primarily is lawn fertilizer and pet waste that gets moving. When we have a rain event and urban soils or soils with a lot of impervious surface, when that water gets moving, it's moving faster. So it's taking more of this stuff with it. And I found a study that had been done in Wilmer. It's really pretty interesting. From 2007 to 2013, they were sampling 2 fields on one farm. One of them was fertilized with inorganic fertilizer and then with turkey manure. The other was not. And then they were also looking at the runoff in downtown Wilmer. So they were catching that in the urban stormwater as it got to a central location. And then they were sampling the water coming out of the drainage in the fields. And what they found is that the urban stormwater contained significantly more ammonium, total suspended solids, and total phosphorus. And phosphorus is, of course, the worst thing in lakes, which is carried on those solids, than the fertilized field, because just the water gets moving faster across impervious surfaces and in urban soils that are not able to bring water in, that are themselves not healthy, that are crusted, that have been allowed to be dry, allowed to be bare. And that it was only nitrate that was less than the fertilized field. But that has to do with nitrate being taken up so quickly by plants in the field during the year. So when we're thinking about our waters and we're thinking about the soil water connection, one of the things is, are we allowing the water to go into the soil and move through the soil before it gets to the waters? Or are we setting our landscape up in such a way that when we have rain or snow, big snow melt events, that there's no place for it to go and it just picks up everything and takes it with it? And these are on sort of bigger scale events here. It doesn't stay here. So this is highlighted is the MOMS. So it's the Missouri, the Ohio, and the Mississippi watershed. You can see Chicago. I'm gonna mark, here's Chicago. There's the Twin Cities. And here we have St. Louis. And there is the Gulf of Mexico. And the Gulf of Mexico has a dead zone fairly regularly. And so I pulled off the EPA website the hypoxic zone for 2021. And it was 4 million acres. And it was a pretty big one. There have been some smaller ones in recent years. The size from year to year can depend on how much flooding there is in the continental United States, whether there's a drought, tends to be smaller. When we have a drought, there's more of a problem when it's bigger, because just think of all that huge area and the amount of soil that's not taking water in, that's not holding on to its nutrients, and then they go down and they dump into the Gulf of Mexico. If you look at the graph, the long-term average is about 14,000 hectares, which is really not very promising when your goal is 5,000 hectares. And this is from 1985, that graph, to 2019, with a lot of states along the Mississippi watershed actively trying to reduce the amount of nitrogen and phosphorus that's getting into their surface waters. I would propose that a lot of the reason that we're not having that success that we want yet on the macro scale is because we aren't taking good enough care of our soil. And so as water is moving across it, instead of going into it, we're just basically taking everything and dumping it in the rivers still. And that we could probably do better if we envisioned our soil as a lively place. Soil health number 4, practice number 4. This is where we get to the buggy person's happy place. Maintain living roots. It's not only how many roots you have in the ground, feeding the biology is the phrase we frequently use. It's how long you have them there. So if you have a lawn, for instance, you know, if you look out in your lawn, pretty soon we're going to see those early perennials coming up. There are things that grow late into the season. Anything that's green and growing is pumping carbon out of the sky and into the soil where we want it. If there are times of the year where you don't have green and growing, then you're basically just telling everything that lives in the soil, oh, hey, you know, you don't need to eat right now. So let's keep this idea of the growing season as extended as we possibly can to be supporting all of the things that live in the soil during the entire year. Because while there are things now that are still basically hibernating in soil, since sap has started to run, the growing season is going, things are waking up. And I'm going to stick this one in just because I finally got to see it last year. Pardon me, I'm a native of Washington, D.C. And I saw the cicadas when they came out when I was 7. And then I missed them every time until last year, and I happened to be there in May. That picture was taken in the front yard of my parents' house. You can see the size of the emergence holes from these 17 years below the soil, drinking root exudates. Didn't bother the tree really at all. You can see them on either side of that tree. Now where that became, I mean, first that was just great. Like, wow, look at all those holes. This is fantastic. How many insects came out of the soil? Billions of them. It's amazing to me. But I was back in August and we had the remains of Hurricane Henry coming through town. My basement was flooding right then. The street was flooding right then. The yard was not flooding right then because the cicadas had done what earthworms also do, and they had created these channels that water could just flow down into the soil. We have annual cicadas here, so this is going on here as well. We just don't have that amazing every 17-year hundreds of millions, if not billions of insects coming out at once to announce their presence. Last soil health practice. This one's a little trickier when you're not on the farm because people think herbivores just mean cattle. Herbivores can be chickens, herbivores can be goats in wetlands, herbivores can be insects. Anything that eats a plant is an herbivore. So if you include herbivores where practical, you're going to increase the cycling of nutrients. We always want to be thinking about that nutrient cycle. And the best example of it really is with cattle. The cattle create cow pats. Within minutes of the cow pat hitting the soil, it will be colonized by dung beetles. The male and the female dung beetle will mate. It's a luxurious hotel suite there in there, the fresh cow pat. And then they will burrow straight down into the soil, taking that cow pat material with them, lay their eggs. Their larvae will eat the cow pat that has been thoughtfully put down in the rooting zone by their parents. And then their frass, which is insect feces, is even more bioavailable. All this stuff is right where the plant roots want it. So you get increased rooting of plants, you get a deeper O horizon, you get a much healthier soil. And this particular soil came from a pasture that I think 10 years previously had basically been lifeless and was turned over into grasses with rotational grazing to allow the cattle to intensively graze one spot and then move. And their intensive grazing, the action of their teeth on the grass causes the grass to spread. So you get more sprouts and more roots. And it becomes a very beneficial cycle. So nibble, recycle, and repeat. And then coming up to the end here, 9 of 10 insects agree. So 90%, we think, of all insects spend at least part of their life cycle in soil. In northern climates like ours, we're usually talking about eggs or pupae. That's 2 stages of an insect's life cycle where it can just kind of hang out for a while. So soil is a great place. You're constant temperature, you're protected. If you're pupating, you're doing the I'm digesting my entire body and building a new one. You don't want anybody to see that. That's ugly. So you want a nice, quiet place like soil. If we are constantly disturbing that soil, this is part of what's happening to insects. We're taking their homes, we're disrupting their eggs, we're hitting them before they're adults and capable of moving away on their own. This particular place is in Fergus Falls. It's a DNR site where they have feeding plots. And for 100 years, from 1887 until when the prairie was broken, that field that you see in front of you had been conventionally farmed. And then the DNR bought it and they planted grasses and prairie forbs and they just left it alone. All of those insects were seen in there. And there were even more. These are just the ones I got pictures of. And we were there on a dynamic soil properties sampling project. So I had 25 soil scientists digging down in the soil and being very excited by the depth of the A horizon and everything else. But then one of our assessments was how many earthworms are in a cubic foot of soil. That's a standard soil health assessment. And they'd be breaking it up and I kept hearing, and what is this? And what is this? And they were seeing all these things that they had not seen before. And then I was getting fascinated by these butterflies because I had not seen that particular butterfly before. And there were so many of them. That is a wood nymph butterfly. So it hangs out in the space right down by the pond where you have trees growing as well. It's an edge specialist. It likes trees on one side and grass on the other. And so I was just making notes on the data sheet of all these different things I was seeing and taking pictures of the ones that I was going to try and identify later. So we have a butterfly that lays its eggs right at the base of the grass. That's where they overwinter. And then going from the butterfly here, we have a staphylinid beetle, a rogue beetle. That's a predator. We had ground nesting bees. We had white grubs, which are scarab beetle larvae. We had a black cutworm for anybody that has ever had their tomatoes or their beans or their baby sweet corn taken out. They're not your best friend. We had click beetles, which can be another pest. And then we had this curculeonid. This is one of the biggest ones I've ever seen. It's 1/2 inch long. These are seed predators. So we had all these different insects that were just there because that space was left alone. They'd found it, they'd colonized it. But then we had two other fields there. One was still a conventional cornfield. We sampled that one second. And the thing that floored everybody is we walked into that field and there was nothing. There weren't butterflies. There weren't bees. There were ants along the field road. There were dragonflies buzzing above because dragonflies, like mosquitoes, that live in corn whorls. But there was nothing. When we went and they dug up the 1 cubic foot of soil to look for earthworms, we weren't finding any earthworms. I was taking pictures of the soil surface, and it took me a while to realize one of those pictures contained a soybean leaf from the previous year. Nobody had chewed it up. There wasn't anybody to chew it up. Soybean litter is high nitrogen. It should go like that. There wasn't anybody to attack it. So the entirety of the recycling of plant material into the soil to get those nutrients going again couldn't happen there because the soil itself was not home to the organisms that would do it. It was a stunning difference. So I'm going to conclude here with this. Soil has an easy button. It's called the soil food web. It only works when it's connected. So it's connected when you follow the soil health principles and make the soil hospitable to the members of the soil food web. So, excuse me. Are there any questions? That's quite a bit in a short time.
Yes, Dr. Journey. This is Jim Betcher. I had a question, something that I saw, wow, I'm going to date myself, 1986. I was living south of St. Louis, Missouri, and it was probably late May, early June, had 6 inches of rain in 90 minutes. Really bad, a lot of bad flooding happened. The ground didn't have time to absorb any water. It just all rushed in. And there was a farmer that had fields on both sides of a 2-lane blacktop highway. And he had just chisel plowed, getting ready to, work the soil, plant it and everything. His chisel plow was 14-inch tines on it. And when this flood came through, it took all the topsoil. You could just see where the top of the tines, 14 inches down. So all of that, we call it good ground, 14 inches worth was gone. Have you done studies? I'm sure studies have been done, but what would you look at as a time frame to get that ground back to the condition it was in?
Somewhere between 700 and 1400 years.
Well, we were taking bets back then. I don't think anybody would have hit that, but because there was a heavy Native American presence in the area at the time, or 200 years prior, and guys that go out and hunt arrowheads and stuff, they're called rock hounds. They were going out and they're just loading their pockets every weekend because all this stuff, it was uncovering history and everything else. But I always thought about that, how long that takes. When you looked, you showed I think your first or second slide, you showed the different layers, and we see that much disappear. And to me, that's, I guess, kind of a marketing point too, to drive it into people's heads. Here you lost 14 inches of soil. You're not going to see it for X number of generations again, that the ground would be in good enough shape to grow crops or whatever. So I've just always been curious about that.
Pardon me. The common agreement is that it takes between 500 to 1,000 years to form 1 inch of soil. But that's starting from the parent material. So I'm going to back up here. And see, parent material. Okay. So on this particular slide, we only got to see, which is the substratum that's decomposing bedrock. So that's the parent material, is the bedrock below that. So if you just take this horizons as semi-representative, 0 to 2 being the O horizon if you had a lot of organic matter there, 2 to 10 inch being the A horizon, and then 10 to 30. So if this chisel plow was going down 14 inches, then he's probably plowing the B horizon. So that subsoil, lower in organic matter. And what farmers have seen over time is that their soil gets lighter as the years go by, because they're losing the O horizon, they're losing the A horizon, and then they're mixing in the more mineral B horizon with that. So it's even worse than just having the topsoil disappear. It's you're mixing less biologically functional soil up every year with what's remaining of your biological hot stuff.
In reality, then the farmer's probably doing damage when he thinks he's doing good.
Well, there is, I mean, I've worked with a lot of farmers over the years, both at the U and then when I was working at USDA. There's a concern when you're this far north, and it's a real concern that you need to have a level, even, equal seed bed. So if you're talking corn and soybean, you need soil that's about 54 degrees Fahrenheit for those, either one of those seeds to sprout. This is one of the reasons that no-till was not adopted for corn very rapidly. It's still lagging in Minnesota because you don't know exactly what depth your seed is going to be at. People were a little bit worried that they might not have the seed at exactly the same depth. And if it's not at exactly the same depth, it might be exposed to slightly different temperatures, slightly different water. It might sprout later than the others. And I've had farmers tell me that a corn plant that's 3 days behind its neighbor is a weed. So there really has been a tremendous effort to make soil as uniform as possible and to get that black soil in the spring when you think the sun is going to warm it up faster because it's black. There's a real emphasis on it. And we've got, you know, I don't know, 100 years of agronomic research talking about making things as uniform as possible. And that is arguing against everything that soil health is telling you, because soil health is, well, let's make it as ununiform as possible. Let's make it diverse. Let's make it active. But the farmers who are adapting to using soil health are finding that it works just fine.
That's interesting. I appreciate it.
Dr. Journey, this is Andrew here. I'm just wondering, as a practical matter, when I see, and we talk about a lot of infill construction in areas where properties might have been developed on either side, but now we're infilling some area that's available for use and someone can build on, but potentially it's not good for the soil. Maybe it's not good for anyone as far as runoff or anything like that. Are there things that we should be looking at to determine whether a property is appropriate for construction? And my second question would be, is there, or has there been a review of any way of reclaiming soils that are runoff? So for instance, if you put a barrier up and you catch some of that soil, can you replace it? Does that become functional soil again or not?
Yeah, if you put a barrier up, you can replace it. But I'll give you a kind of a little example of this, and it comes from my own yard. Last summer last fall, I did a talk at Minnetonka, and I could dig up some soil from my yard at that point. And I had 3 different samples. I had one from my tomato bed, which is very healthy soil, and I don't turn it. And I stopped turning it when I got the job with USDA because I thought that would be hypocritical of me, even though I didn't use a rototiller. I used a fork. I have front yard. I'm here in Southwest Minneapolis, so I have a tiny little front yard, It gets road salt. It's had stuff piled on it. It's compacted. It's awful. And then I had a side bed that had been in perennials and left alone for over 20 years. But we had a water problem in the basement, and my husband and one of my sons decided that, you know, kind of putting a little drain system in there might be a good idea. So they dug it up. They were very careful. They separated by color, and then they tried to put it back in the right order and everything. And it's wrecked. It's just wrecked. But the degree to which it was wrecked, I mean, it was bare, so it has crust on it now, but the degree to which that disturbance had changed it was not apparent until I did what's called a slake test. So I took a clod and I let it dry out completely. And then I put that clod in water on a screen. And I had one from the front yard, which is very unhealthy soil, and one from the tomato bed, which is very healthy soil. And then I had the one on the side that I thought, okay, well, it was healthy soil and it was disturbed. And the one from the front yard, because it's not lively, it's not well glued together, it just kind of slumped in the water. It didn't explode. Sometimes they do that. But as the water rushed into the soil, it just kind of collapsed. The one from the tomato bed just sat there and went, hey, I'm a sponge. I can take water all day. And the one from the side where it had been disturbed broke apart. And the parts had been put back, but the structure that used to hold the parts together wasn't there yet. So what I will have to do is throw some prairie plant seeds in there, get some native plants in there, plant it up, and then leave it alone. Now, I don't know if I'm still going to own this house in 30 years, but if I leave it alone for 5, I'll probably see that it will be in much better shape than it is now. So when we're doing infill construction, I'm not a soil scientist. I don't do soil engineering. I am an ecologist, and what I would say on infill is when I mentioned the impervious surfaces question and the transport of materials from the soil into our waters, when we look at landscapes, sometimes it's probably not a good idea to build on slopes. You know, if we have a slope and we're going to disturb that, so that thinking about my side yard was disturbed. Now it is not as strong as it was. So you could come and you could pound that soil together and firm it up, but that would be just compacting it to a point of some stability. Whereas before it had structure. So when we're thinking about this and we have a patchwork of a landscape, there's impervious surface over here, there's concrete, there's roof. These are all places where water can't infiltrate. If we have some patches where we can let it infiltrate, that's probably a good thing. I hope that answers your question in some way.
So I have a question if nobody else wants to go. So Ann, this is Jill Crafton. So
hi Jill, recognize your voice.
Okay, it's good to hear you. So I've been on this watershed board since 2009. First 3 years were kind of dysfunctional, to say the least, but we've turned stuff around. We're making good progress. We're doing really good things. But the focus in the watershed world and what I'm seeing even at Bowser is we're focused on managing volume, rate, and velocity rates. And I mean, we're not solving the problem. We keep diverting things through wetlands into culverts and down to the Gulf. And it just seems like there's this pattern. And that's why once I was at that Nobel conference in 2018, it was like, oh my gosh, you know, we're doing everything backwards. If we really care about water quality, then we have to understand how the water cycle works and that it's part of infiltration and being taken up by the plants and then transpired out through trees or whatever. Liz was introduced when she first came on. They were doing a thing on, well, what's a watershed? And it was like, well, it's where water drains. Well, we've gone from like 10% runoff to 55% in urban areas. So maybe we need to be looking at what the water's traveling over and what condition that's in and why there's no functionality there to actually do the infiltration. So we're grappling with how do we take soil health principles and practices and knowledge and move from just moving the water to actually restoring a healthy water cycle. To me, that's key. So if you got any tips on what we should start doing, I know like Gabe Brown talks about using 16 species in his cover crops so we can build soil. But I don't know what we can do in an urban area particularly.
If you have 1 square foot, you can do something. I mean, literally any little space that is not covered, that's ground, there's soil there. So if you have these not covered spaces and you're thinking about the life of soil, all the things that are working together in soil, if you're trying to make that a more active space, if you're thinking about having more things living in it, not on vast scale, on micro scale, just in your yard. I'll give you a for instance. I mean, the immediate effects are right there. So 4 years ago, I stopped turning my tomato bed, as I mentioned. And it was pretty good soil. I dumped a lot of kitchen scraps in there and been feeding the worms. But I used to have to water my tomatoes a lot. And I have a tiny yard. The only thing worth growing is tomatoes. And I had to babysit those tomatoes on hot days. I'd watch them wilt. And I'd watch the fruit drop. It was depressing. And I got blossom end rot all the time. Now, this is a small space. It is maybe 15 feet by, no, it's not that big. Maybe 10 feet by 3 feet. And so I stopped turning the bed. And then I got even lazier and I stopped worrying about the weeds. They're my cover crops. I don't know what they are. I haven't identified them. I've got some grass, I've got some dandelions, I've got a few other things, I got purslane, but there's a whole bunch of other stuff in there that is just doing its thing. When I go to put my tomatoes in, I weed whack them, dig a little hole, put in the tomato, close it up, ignore them. So last summer, when we had watering restrictions in Minneapolis, I wasn't watering my tomatoes because, you know, watering restrictions. That was the time of year when they were starting to set fruit and I had beefsteaks. I wanted to get those big fruits. And I went out one day and I'm, you know, going to tote the watering can out from the house. And I looked and all the leaves were fine. The leaves are out. They're photosynthesizing. They're doing their happy thing. My grass is dying, but my tomatoes are just fine because I had healthier soil under those tomatoes. That is a very small area. So if you think about all the little small areas in urban spaces and see them all as opportunities to make healthier soil, to have better places for pollinators in urban areas, to have, you know, all the things that we think about that we would like, you know, look out, see flowers, butterflies, all that stuff. The scale does not have to be large, but the impact can be, even at small scale.
Would you advise doing a little bit of soil testing to find out what the heck we've got? Because so much of urban area, if we're going to try to encourage projects, we don't know the condition of that soil biology anymore.
Well, it's in urban areas, I'm less worried about the condition of the soil biology as I am what contaminants might be present in the soil. I mean, particularly for things that people are going to be eating. There's a little bit of faith in not having too much horizontal transport in my backyard. I know my tomato bed's all right, but other parts of my yard are not so friendly because people used to bury their garbage in their backyards. In this part of town, my house was built in 1914. So I would say in an urban area, to know that your soil is safe is an important thing. But to look at how it might be functioning, if you take a soil sample and you go to a lab, the lab can tell you several things. It can tell you how much of various nutrients are in the soil right then. It cannot tell you if they're being used. It can tell you what the bacterial fungal ratio is. You could even have your soil sequenced and find out the DNA in the soil. But how they're all working together or not, what they're actually doing, a static test can't tell you. But watching the things living in the soil, accessing the soil, can tell you quite a bit. So if you want to know how biologically active your soil is in terms of the decomposers, bury a pair of underwear.
Okay.
It's called the whitey tighty test. You know, you take your white cotton underwear, you've got the, it can't be bleached now, that's not fair. It needs to be either unbleached or it needs to be new. And you bury it in the soil, you leave the waistband up so you can find it again, bury it, 3-4 inches deep, and come back in 3 weeks, 4 weeks, however long you want to, and see how much of the underwear is left. Because white cotton underwear is food to soil microorganisms. So if you give them food and they are there and they are active, they will eat it. And in a really biologically active soil, you could try to pull on that waistband and just have it come out in your hand and not much left.
Okay, so you've talked about observation before, using just observation. So then even like the insects, birds, and other parts of the ecosystem might be helpful.
Well, they're telling you. I mean, when you see insects in the soil, they're telling you they can live there. If you see birds hunting earthworms in your yard, they're telling you there's earthworms there. They don't have time to waste. They're going where the resources are. So if you're paying attention to things that are big enough to see, then you're already learning a lot about the condition of an environment and of the soil environment. And then this last week, the people that I was working with at the U on the Soil Health Initiative, we just got our manuscript revised and in. And what was interesting, we did 42 different measures of soil health, some of them in the field, some of them in the lab. And the interesting thing was, in comparing the non-cover crop and the cover crop field on these farms, the only things that came up significantly different were earthworm counts, the soil smell. Does it have that wonderful soil aroma, or does it smell like chemicals? Infiltration was close, but that can be hard to get a good measurement on. There was one soil enzyme that came up. And the VES, the visual examination of soil structure. So if you're curious about what the VES test is, I have a YouTube up where I did one for the Pollinator Summit. So if you go on YouTube and you search Anne-Marie Journey, you'll find me. Unfortunately, usually without a head and with way too much of my not in shape body on the screen. It was painful to edit, I got to tell you. But I broke down soil and I talked about the visual examination of soil structure. That's a test that was developed in Great Britain. And if you think about the gardening culture of Great Britain, they pay a lot of fine attention to their soil. And this is just a wonderful way of looking at soil and evaluating it. You can do it yourself.
Okay. I looked for you on YouTube and something had been taken down. So
I was able to find it. If you put pollinator next to Anne Marie Journey, you can find it.
okay.
Or soil. Yeah, I had watched it during the original Pollinator Friendly Alliance conference, and I rewatched it just earlier today. So it's there.
Okay.
Can you tell me, if we still have time, you talk about soil function, and I think of soil as filtering water, infiltrating water to replenish the aquifer. I think of it more recently in terms of carbon sequestration. Can you tell me some of the other functions of healthy soil?
Well,
I know
you got quite some of the headline ones right there, but if you think about what we as humans expect soil to do, and that's kind of how we approach soil health. So a healthy soil is one that a plant can stand up in. So for a plant to stand up in soil, the roots have to be able to grip aggregates, and it's actually the root hairs that do that. Those aggregates are created by the things that live in soil. So the root hair helps in doing it. The mycorrhizal fungi help in doing it. The microbes help in doing it. They're creating the soil glues that are providing a stable matrix that something as big as a sequoia can stand up in. If it's not healthy, it'll fall. If the soil isn't healthy, the plant's going to fall over because it won't have that structure to physically hold itself up. A healthy soil is one that, as you said, filters water. It also can filter air, because remember, when water gets down into the soil, it's pulling air with it, not just oxygen, air. So that air is moving through the soil pores, and it's being acted upon by the things that live in the soil. So soil is a place where things go to live. It's a place where dead things get recycled. So going back to my example of the dung beetle, if we did not have that mechanism by which beetles take poop and put it down in the ground and live on it, we'd all be hip deep in dinosaur dew and everybody else's dew that had been here for all of these years, because soil is this matrix where all of this recycling happens. So if it's, when we're thinking about carbon sequestration, one of the things we're trying to do is bring the carbon back that we have liberated through large scale, conventional agriculture. Pardon me, my allergies are starting up. In some parts of the world, we estimate that half of the carbon that used to be in the soil is now somewhere else. So that would be in two big sinks. One would be the atmosphere, and the other would be the ocean. And in both of those places, carbon is not doing its job. We want it in the soil. We want it to grab water and hold on to water and let water be available on an as needed basis, instead of just when we put it there because the water's running through so quickly in a denuded soil. That's just one of the things that we're asking it to do. So when we think about it, I learned all this stuff after, no kidding, 20 years of working in cornfields with an insect that lived in soil, doing my PhD with a nematode that I would put on the soil to get in the soil to control the insect, and never once seriously thought about the condition of the soil itself. Neither did my committee, which is kind of embarrassing for all of us. But I had that understanding after I started to learn about soil health. But I did my PhD in the 90s. Before this was a big movement, a big idea.
Mm-hmm.
So yeah. I plead ignorance of time.
Yeah. Yeah, the other thing I want to ask about, thank you for that great answer. I think the more we learn about the soil, the more important it is to keep it healthy. You said something about life in the soil is all within an inch of a root. I thought I've seen pictures of those where you got the roots of the plant and then the mycorrhizae fungi extend that, you know, for many, many more feet.
Okay, so going to the two clods, the rhizosphere is named for its close relationship to the root itself. So if you think of the root as being the primary source of energy to the soil, because all of these biological processes are ultimately being powered by the sun, because the sun is providing the energy that the plant uses to take carbon dioxide and water and create sugar. So that sunlight doesn't get down into the soil to activate things in the soil to photosynthesize, except algae, and they're very close to the top. So the plant is transferring that via its roots into the soil. So if you just think about the simple process of diffusion, you know, you see a drop of coffee on a napkin spreading out. And that's a poor model because it's nowhere near complicated enough to explain all the different processes that are going on. But that would be kind of the model mentally for the idea of the rhizosphere, that the root exudates come out of the root and they spread only so far, and then they're used up. And so that the more roots you have in the soil, the more of the soil is in a rhizosphere. So fungi are depending on carbon. So if you see all those white fingery things in that picture, the fungal hyphae are essentially looking for carbon that a plant put there. So the plant is the way that the carbon gets in the soil. So if you think about if the root focus gives you that understanding that this is imported energy. Does that help?
Yeah, it's just fascinating. Thank you.
If anyone else has questions, we can certainly get them to Dr. Journey. But I'd like to thank you very much. This was a really interesting presentation and I think a great discussion afterwards. So thank you very much for attending. Appreciate it.
Oh, my pleasure. Thank you.
All right. Yeah, thanks for joining us.
Yeah, thank you very much, Doctor.
I give her a round.
Yay.
Thank you very much.